Okay, welcome back, everybody. Again, my name is Robert Slowinski. I am a biologist, genetic genealogist, and we're going to cover a presentation I call Navigating Paternal and Maternal DNA Results. For those people who have done those kind of results, this may be a helpful walkthrough. If you have not, this will potentially show you those might be useful for your genealogical research. Let me minimize this and let's get going. Okay, before we covered a similar slide that shows the branches of the family tree. Basically, what lineages are followed with YDNA or mtDNA? Let's recap that. Let me get my laser pointer. Here it is. Showing that the male line is one long thread along the paternal line, showing the red boxes on the left-hand side, and that the maternal line or mitochondrial DNA line follows one long, thin, linear line down the right-hand side with the diamonds over here. That's just a recap to make sure that if you did see the previous presentation, you'll already know. If you haven't, you're just logging in right now. This kind of gives you an idea what we're going to be looking at specifically regarding DNA testing for maternal and paternal DNA. Not to be confused with paternity tests, which is totally different. Okay. The DNA that is tested, YDNA, DNA, where is it from in your cell? Think of a cell as an egg cracked open, frying on a frying pan, and you have essentially the YDNA comes from the nucleus of the cell in the center. The Y chromosome is passed on from generation to generation for men. And the mitochondrial DNA is located outside the nucleus, and it is found in the mitochondria. Otherwise, I call them the kielbasa's or Polish sausages of the cell. They create energy taken from energy that the food from you, what you eat. Let me see here. Let's move on. Okay. Paternal DNA is based on the Y chromosome, and we'll be looking at results for STR markers and SNPs, and I'll go into more detail about what those are. It is basically one chromosome for each cell located in the nucleus of the cell. This is an image that shows myself, my father, Tadeusz or Tad, and his father, Alexander Slowinski. Showing that essentially that the Y chromosome is passed from Alexander to Tadeusz to me, pretty much intact. Very few mutations occur between generations. So that allows you to go back through time, and with the number of changes that you see, you can determine how far back you have a common ancestor. On the paternal line, and that paternal line usually follows a surname, but not always. Okay, if you want to do any type of paternal or maternal testing, the testing company to go to now, and it used to be other companies did this as well, but now the only company that I trust regarding this is Family Tree DNA. They do the YDNA tests with the STR markers. Now, STR stands for Standard Tandem Repeat. And I'll repeat this a little bit later and I'll explain what's going on with that. There are the marker tests that you would look at is the basic one would be 37 markers. Then it goes up to 67 markers and then a 111 markers. Now, it doesn't really give you that much information at 111 markers compared to like the big Y, which is like 500 markers. But the 37 markers, 67 markers are good starts to find out if you have a match, are they closely related to you or not? And you can always go up a level to see if that holds true. There was a question about costs or price associated with these tests. And as we took a break, I completed this entire presentation within 15 minutes. I amended a couple of things, but I wanted to show some costs that are like hot off the press for paternal ancestry, the YDNA, $100, family ancestry, $59, and that's the cousins test. mtDNA would be $139. I'm not promoting this. There was a question, and I thought people should know. So the mtDNA tests, or mitochondrial or maternal tests, include the HPR1 and 2 plus the full sequence. At this point, I think they're only offering the full sequence. It seems to be about the right price for that. It used to be close to $300 a number of years ago, back in like 2007 when I started this, and has appreciably fallen. Not as much as the family ancestry one, but still very good for costs. And some people give these as presents for other people for Christmas or other holidays. Okay, family tree DNA test kit looks a little bit like this. There are some companies that have a spittoon or a spit vial. You basically you don't drool into it, but you actually gather up enough saliva. And the first time I did a saliva test, I said, well, that's no problem to put that little amount in that vial. Boy, it's a bad day to you have a dry mouth. So the idea before you take any of the DNA tests is not to eat or drink anything at least 20 minutes or longer before you take that test. This is a cheek swab. It allows you to scrape the inside of your mouth on the inside of your cheeks. It is a very soft brush, almost like a paperish bristles on the end of it, and they lightly, gently scrape the sides of the inside of the mouth for about 30 seconds. You push the tip. Let me get my laser pointer out. This tip essentially goes into the vial. There's a solution in there to preserve it. And when you are ready to do that, you eject it by pushing the plunger, and that pops the tip into the container. You take 2 samples. They could be the same day or the next day. If you do one the same day, do a one side of the cheek and then the other cheek on the inside for the other vial. They do it so they have enough DNA tests to test in case the first batch does not run properly. There are times when there's a problem with the processing of the DNA to get it to go through their sequencing machine. And so what they do is they have to have a backup. And that backup can be stored for up to 25 years if you wish. If you tell them, you can check mark basically that you wish to have that sample after it's been run to be destroyed. So that's not a problem. But if you keep it, or let's say an elderly relative that you have, you may want to have it later on for additional testing to help with family tree investigations, family tree research. So let me see here. I think that's it for this one. Okay, when you get your YDNA, and you're probably looking at this going overwhelmed. I see a bunch of numbers. I see a bunch of letters. I don't know what is going on with this results page for YDNA. And these are panels 1 through 4, and each panel has a number of marker names. So let's start out with the first one. It's DYS 393. That again is an address, like 8901 North Oketo, would be the address of a location on the Y chromosome. Now, the other one I gave you was a real address. So the address that you have for that YDNA has a marker value, and that value is the standard tandem repeat. How many repetitions of, let's say, an example, these are bases, C, C, G, G, A, A, T, T, cytosine, guanine, adenine, thymine. These are the base pairs, the 4 bases that make up your DNA. DNA is only made up of 4 base pairs, and that the way they're connected together and what arrangements they are provide basically and make who you are you. CCGGAATT. Now, this is a segment or a tandem. Repeat is the number of times this section is repeated. So this section at that marker is repeated 13 times. This is just an example. This is not a real number of a base pair order, but it gives you an idea that a segment of DNA repeats itself or is repeated 13 times at this location. At this location, at DSY 390, it's repeated 25 times. And so if you look across the board, and these are my results, you then can compare your marker values to someone else's marker values. And we'll go over that a little bit later, comparing marker values to see how closely on the paternal line you may be related to someone. So initially they started with a 12 marker test, and at that point you're related to almost every man in the world because it's so generalized, and a lot of people have the same marker values. 25 is going up, but now we have 37, 67, 111, over 500. But this gives you an idea of what the marker value will look like. And it might look like just a bunch of numbers, but after you've looked at the number of these, you start then comparing them to other males that have the white test to see how many differences you have. Okay, these are paternal matches at 12 markers. Now, if you can see, the balloon right there goes to my white, he's hiding in there. That's my distant ancestor, is Alexander Slowinski, born in 1898 in a town called Grzymkowice, Poland. Now, I do speak Polish and English. I was born in this country. My parents emigrated from Poland after World War II to seek out a better life, became very productive people, obtained their citizenship legally, and we just had my brother and I as children, but my father was a mechanical engineer, and my wife was a nurse, and my mother was a nurse and then became a hairstylist. So these are the exact matches and one-step matches at 12 markers. You notice that I have some in India, as well as Central Asia, and a lot in Europe. The 12 marker test kind of gives you an idea of what generic haplogroup you belong to. And this is the Indo-European group. You can see the Indo-Partner-European that is one of the largest genetic groups in the world because it spread out so wide, far and wide. R1A and the Indo-Europeans were the ones that, according to what I understand, domesticated the horse and invented the wheel and wheels, wagons, and chariots, and were very mobile during the Bronze Age, some several thousand years ago. So the next one here is probably 25 markers. You're seeing a lot less. The higher resolution you go, the less matches you get. And these are my matches at 67 markers. This is a match page that you get, gives you an idea. The names, I have very few. Genetic distance of 3 at 67 markers. That kind of gives you an idea that my match with a gentleman that lives in the United States, originally from Russia, has a distance of 3, and I calculated using the tip method, approximately we had a common ancestor in the 1700s. His genealogical tree does not go as far back, either does mine, and we're still doing research to see if we can get back that far to find that common ancestor. So it shows essentially in this bottom box here that you can email that person, that you can figure out the time to most common recent ancestor. You can write some notes down for yourself. This is the test, the highest level test that they took. And they also took a family finder or autosomal test in addition to the YDNA test over here. This YDNA haplogroup shows the final or the end mutation that is on the branch, whereas the last one here, M173, is much further down on the tree, but the resolution on their test may not have been as high as the ones I've taken and the other people have taken as well. Okay, let's talk about the YDNA tip report. Okay, you're comparing 67 markers to 2 people, and let's focus on this. At 4 generations, it's a 51.74% chance that we have a common ancestor. Now, I know for the last 3 generations, and I put that down here, that we're not have any common ancestors. So that 67 markers and display every 4 generations. We know that at 8 generations, we have a 91% chance that we have a common ancestor. That's 240 years ago. Now, that comes out to the late 1700s. You might say 8 generations. Well, how many years are you accounting for generations? Typical generations that I've seen mark down pretty much every 25 years. However, I have been looking at the some family trees and noticing that the men are getting married a lot later than 25 years old. And some of them are getting married at 30 years old, even back in the 1700s, 1800s. So I'm basically averaging between 25 and 30 years for the time frame for a generation. And when you get to 24 generations, we have a 100% chance of having a common ancestor. As soon as it hits the 90% range, that's where you can figure out, this is where you can start focusing on when we can possibly find a common ancestor, even though it might be further back than that. But this is a tool that you can use at Family Tree DNA to come up with a possible time frame for when you have a common ancestor. That covers the STR marker tests, and we are going to look really quickly at this point at the big Y test, and those look at SNPs. SNPs stand for single nucleotide polymorphism. And you're running away from the computer as I speak, I'm sure. What it is just one change of one base at one location. But they had to come up with a fancy name anyway. So at an address of, let's say, the location 26940, there is a base pair change, letter change of the DNA, and that kind of gives you an idea, because its bases change relatively slowly, that you can look at those and determine by comparing it to other men, their base pairs, how closely or how distantly they are related to each other. The SNP tree covers the entire genetic tree for all males globally. Regarding this, essentially, there are, at this level or branch, Y33, I have 30 matches. When you go to a more refined branch, I have 30 matches. And finally, the refined branch of 24958, I have myself and 3 other matches that are at that location, or at that part of the twig at the end of the branch. The other thing I found out is that I have a private mutation, an SNP called L450. And anybody else that has the other mutations that I have in my genetic tree and has L450 is definitely directly related to me. Now, the gentleman that I was more closely related to with the STRs in the YDNA test, I asked him if he would take a specific L450 test in addition to other ones. And he did that, and he did not have the L450, which means that the L450 probably arose in my genetics, my family, between the 1700s and the 1900s. So it is a relatively new mutation within my genetics. If I ever find somebody that has the L450, aside from close family members, I will know that it's probably within that time frame, and then we are definitely related. It kind of gives you an idea that private mutation can give you an indication of when potentially that mutation may have occurred if you've compared to other people that have tested, and giving you a time frame for a common ancestor as well. Okay, now, if you start at the top of the tree, the main trunk, you can essentially look down as you go down this SNP tree to look for the different mutations and branches. That kind of gives you an idea. Is it useful for genealogical research? Yes and no. The yes part is that if you've done DNA testing using the normal DNA STR tests, you will then find out your matches. If you want to confirm a true genetic tie to that person, you will ask them to test the SNP, that branch or twig that you are done or finished at, and see if they match that SNP or that single mutation. If they do, so you then packaged the marker test with the SNP test together, and you have confirmed a genetic relationship at that point. If you have family trees that can now be combined, you can do so. Or if there's still a couple of branches you need to research, that still gives you the knowledge that you are definitely related. You're just going to find that common ancestor and do some more genealogical research. So that's how it ties in. Okay, I've talked a little bit about haplogroups, and this looks like a very crazy map, but what it is a map that generalizes the locations where certain male genetic groups are found globally. And I mentioned a little bit about R1As, which one I belong to. I know there's a person that I will talk later on that is related to the R1B group over here. Both R1A and R1B originated in Central Asia and broke off and split and moved into Europe. And part of R1A broke off in Central Asia and moved down into Northern India. But there are genetic groups all around the world, including E3B2, which is a North African group, as well as groups like the Aboriginals, which are C4 and K4, Native American groups, which based on genetics have been determined to have come from northeastern Asia and migrated tens of thousands of, well, let's say 14 to 17,000 years ago into across the Beringia or Bering Strait. That was a landmass at one point, and into North America. The haplogroups associated with that are C3b, Q3. There is apparently some R1b that snuck in there as well, but Q3 and Q3 is a dominant group that went into southern North America, central, in the Central America down to South America, and is a dominant Native American male haplogroup called Q3. If you are from the southern United States, in the area of Mexico, Central America, if you do not have a Q or C male genetic group, that means you may have actually a European R1b group or several other groups that were associated that were essentially people that migrated into the area and settled into the population. That is always possible. But you never know what male genetic group you are in for men until you test. Let me see here. Okay, in Europe, the dominant groups, and I know there tends to be a heavily European pushed or toward genetics results, primarily because initially many people with European ancestry wanted to test. And National Geographic has gone globally to get indigenous populations to test as well. So they have a better idea of what's going on globally. And that's where that map came from before. But in Europe, R1A is predominantly in Eastern Europe, R1B in Western Europe. There are a mixing of these groups as well, but these are the dominants in there. I2 is in Southern Europe, whereas I1, the Nordic or Viking basically type of genetic groups are in the Nordic areas. There's an N group in Finland and Lithuania as well in that area. E basically focused on is in Africa. J2, J1, the groups down in the Middle East. G is a smaller group that is in the Caucasus of the southern Russia. And then we have C3, which is more of an Asian group as well. But this kind of gives you an idea that these are the areas in Europe. Now, there, as I showed you the global map before, and I belong to the R1A group. Okay. I am administrator, a volunteer administrator of the Slowinski Surname DNA project at Family Tree DNA. And it is a Polish surname pronounced Slowinski. And you're probably wondering why there are plums associated with this image over here. The surname Slowinski means from the place of plum orchards, literally. But there are Slowinskis that are all over the world, including not only limited to Poland, Germany, Denmark, France, United States, but they're also in Argentina and Australia. And as of the 2002 census, made it up that day, there are approximately 9,000 Slowinskis in Poland. It is not a super common name in the country as compared to some other ones like Novak would be very common. But I wanted to start this page to allow other people with the same surname to join to see if we're related, because we have a common surname. But that's not always the case. So I wanted to branch out to get more people to join this Slowinski DNA testing website at Family Tree DNA. So I started, a Slowinski Foundation Facebook page was started by a gentleman in Australia. And once I started posting a couple times to it, he gave me the administrative rights right away and then jumped ship. So I decided to ask a number of people that had the Slowinski surname to I was providing them with DNA tests so they could determine if we are related and how closely we are related to each other. This started a number of years ago, and I got a number of takers. And this is kind of what remember we talked about the STRs before? Let's focus on the bottom of the page over here. We have Slowinskis and a couple other people that joined as well, maybe variants of the name. But the idea is that you're comparing the marker, let's say the 13 and 25. So all these people have right here, the men have the same 13 and 25, but there's some variation between the next marker, 15s and 17s, and the next marker, 10s and 11s and 12s. So you see there are some differences between the men that have a Slowinski surname or a similar surname, or ones that have had their name changed. So I compared those together, and we came up with this graphic. Okay, so we have a number of Slowinskis that tested, but we're not all related to each other, at least not closely in some cases. But I found out that there are 3 main genetic groups or haplogroups that Slowinskis that have tested in the group belong to. We have the bashful Richard here, Slowinski, who belongs to the Southern European genetic group I2A. Southern European group originates in Southern Europe, and this distribution map shows how they've expanded throughout Europe, and that low levels are found in Western Europe, but the highest levels or numbers are found in Southern Europe. The I1A group, consisting of Arthur, Robbie, and Paul, are a Nordic group that basically the box shows where the ancestral origin of Poland is, but that the Nordic group or the Viking group is found predominantly, the highest numbers are found in Norway and Sweden and Denmark. The group including Robbie, Stefan, and myself. Stefan is in England. Both Robbie and I Robbie's in the Chicagoland area, and I was in the Chicagoland area at the time. We are part of the R1A group, which is mostly in Eastern Europe. So although we share the same surname, we are found also in genetic groups that are different from each other, that have different origins, and are part of the genetic family tree, but go further back. I found out that myself and Robbie, who's a friend of mine, have a common ancestor 4,000 years ago based on our genetic differences. And you're probably thinking, how is this useful for genealogical research? In this point, we were trying to find out if we were cousins that were close or distant. We do share a common ancestor a very long time ago. And for an instance that I have a story where I did find that I was genetically related to very, very distantly to a gentleman that was originally from India. Remember I showed you that map that showed there's some locations in India that had that. Now, these are not like a group of gypsies that came from Europe, went down to India. This is actually part of the breakup that happened some 3,000 years ago, and found out that we are genetically tied to a common ancestor from 3,000 years ago. And so an interesting thing to find out, is it genetically relevant? Genealogically relevant? Not exactly, but it gives you an idea that we have common ties with other people distantly that make the world a lot smaller. Let's move on to maternal DNA. The graphic that I showed earlier, the photo of my great grandmother, Paulina, and her daughter, Wanda, my mother, Apollonia, and myself. Now, my mother passed away a couple years ago. When she saw this presentation, I first came up with actually, this is part of a publication back in 2010 that I published in a magazine. And she asked me, Apollonia, my mother asked me, where did you get that picture of me? I said, I found it in some old photographs. She goes, that's the only time I had my hair dyed black. And I'm like, well, it was a very nice picture. She goes, and she's blonde. She said, I don't know why I even did that. It was just a whim, and I never did it again. And you happened to put that picture in a publication. I said, I'm sorry, Mom. But she goes, you did a good job anyway. I'm very proud of you. So I was happy to hear that. Mitochondrial DNA, again, outside of the nucleus, in the cytoplasm. And think of mitochondrial DNA as being a ring. It's a very small ring. And the areas that majority of the time they look at for genealogical or genetic genealogy is HVR1 and HVR2. Think of this as like a diamond ring with the diamond on the top, the two hypervariable regions as the crown over here. That's for the basic and moderate level test, whereas the full test looks the entire ring that goes all the way around. And that's the rest of it is called the coding region. The hypervariable region, this area right here where the crown is, mutations occur relatively regularly or more often than the remaining coding region. And those mutations in HVR1 and HVR2 are benign. And geneticists were able to find that they can compare maternal lines of many different men and women and determine by this location to determine a maternal family tree. Now, the best comparisons are when you do a full genetic sequence of the maternal DNA, and that includes the coding region all the way around here. Now, mutations in the coding regions, the mtDNA is used for processing of energy they're the power plants of the cell. Mutations in the coding regions can be not benign or potentially fatal. These are some things that you do not grow up with. Essentially, you may have a mutation in the coding region that has implications for some genetic diseases, but there are some that are fatal that just don't make it to cell formation to create a person, essentially. But those people, majority of people do not have any mutations in the coding region. Those are very rare. But the mutations that happen in HVR1 and HVR2 are much more common and benign. And so they're able to use that for determining the maternal family tree. mtDNA is also the most common DNA found and the most abundant DNA found in the body. So that when people, ancient remains are found, that is the easiest DNA to find the most quickly to determine what their genetic group that they belong to. And when ancient remains are found, that's what typically they do. YDNA suffers a little bit of breakdown regarding its lasting over time, but and is less abundant, but there are now new methods to be able to amplify the DNA to get it to the point where they can read it and then get some valuable information from it. But it is passed down, the mtDNA is passed down from the mother to all her children, not just men or not just women, to all the children, both men and women. My great-grandmother, grandmother, mother, and myself, passing the mtDNA down through the lineages. So my brother has the same mtDNA as myself. Okay. I belong to the genetic group, the maternal genetic group, H2A2A1. And that is part of a group. H2A originated in Central Asia and settled down into the Middle East and branched out from the Middle East into Eastern Europe and Western Europe. And a big portion of H2A2 settled in Northwestern Europe. And H2A2A1 happens also to be the genetic group maternal genetic group that was the first mtDNA that was sequenced back in 1981 by Anderson. And he basically there was a lady in England that donated her cheek cells to come up with the mtDNA, and they did a full sequence on that, and it happened to belong to H2A2A1. Historically, was a big deal because after that genetic sequence, all other mtDNA sequences or maternal sequences were compared to that sequence. I am this somewhat kind of closely related to that one woman in England that donated her mtDNA for that study. Since then, the comparison to the maternal genetic material has gone to a more African derived genetic group, so that it is more based on original geography of where our mankind in general, historically, archaeologically has come from, rather than the comparison to this just one lady's maternal DNA from Europe. This, as I said before, I have provided my maternal DNA for research, for different research projects. And I do have an international number where my genetics, my DNA is for my maternal DNA. Again, it's only a very small amount of my DNA from my whole genome. And that has been used in the creation of the RS values. These are a more recent value system where they now compare all other maternal lines to. So they use my mtDNA in part of that study to come up with this new system to compare people all globally. So when I talked about HVR1 before and HVR2, these are the different locations or the results you might get from that. And then these are coding regions results. Now, all you're seeing is essentially letters and numbers, and like, what does that mean? What it means essentially is that the base pair G and A, these are the connections for the base pairs, and this is the location, the 709, where that's located. This right here, G73073R, means that I have what's called a heteroplasmy. And heteroplasmy is just that I have two slightly different kinds of mtDNA or maternal DNA in my body. That means I'm in the process of some of my cells changing or mutating by one base pair. So that when we said, when you talk about how many differences are you from another person, I'm part of my DNA in my part of my cells is in the process of changing just a little bit. And so how is that significant? It's a benign mutation. It doesn't really mean much except for the fact that if you're comparing your genetics to another person, it's important to keep that in mind, that some of it's going to be a little bit different by one. And again, if that's one off, it could be over 200 years since you've had a common ancestor. But in my evaluations, I discount that. A famous person that had, or family that had a heteroplasmy or slight change in there was the Romanov family, the Tsars of Russia. And it's not limited to just people in Eastern Europe. It's globally essentially that this happens. But when they were trying to compare the Tsar Russias to other DNA that was found from their subsequent execution to try and match up the family members, they found out basically that one of them had differences, and they basically discounted some family members of it. What they figured out later on is that there was the heteroplasmy, or just a slight mutation, because that person at that point had two slightly different versions of the same mtDNA in their body. It's a little bit rare, but it's something very interesting to talk about. Okay, mitochondrial results. Initially, when I have a 0, that means that they've done something wrong so far, because I've not had anybody less than 2 differences away from me. And again, at 2, that could be the 400 to 600 year difference in genealogically speaking. But it kind of gives you an idea. This is what results will insert with and surname. And you can do different types of HVR1, HVR2 coding region total to and determine the number of matches you want to show on that. So that kind of just gives you an idea what a results page look like. Again, this shows whether it's a full mitochondrial sequence. The FF is if they've done Family Finder, and you can also check if they have a blue box, a little tree on that. That means they actually have their family tree also uploaded as in addition to their other tests. And you can search their family tree and run checks on surnames as well. So you kind of see this before. Matches HVR1 pretty much globally. A lot of people I match. HVR2, I'm the white balloon again. Shows most of this is a Northwestern European or Northern European genetic group. And this is my full genetic sequence. This is the basically, and none of them are exact matches. In fact, the closest one is a 2-step found in Poland, found in Sweden, Norway, Ireland, and in England. And I do have a 3-step match that I've made a contact with in Northern Ireland. And we'll talk a little bit more about that on that paper that I prepared on mtDNA. But you can still see that at a full genetic sequence, that my maternal genetic group is very Northern and Northwestern European, which means to say, how did my maternal line get there? That is my mystery right now. Okay. Let's talk a little bit about contacting matches, because some people want to contact matches, and there may be some issues with that. Because not everybody wants to be contacted. They probably don't know you. And they probably think you want money or something like that. So when you email somebody the first time, be personable but not excited. Don't start off writing to say that they are your cousin. Because, you know, most likely genetically they are. The question is, you have to also accept the fact that you might be someone else's cousin at the same time. But I tell you what, let's settle this down a little bit and just ask if they feel comfortable sharing family tree information, and be upfront with that kind of sharing as well. But know that not everybody, first of all, will answer your email, but every once in a while, follow up with that, but also follow other leads as well, and see if you can find a person that might be interested in talking to you, not only about your DNA results, how you shared with them as cousins, but also if they're willing to share some interesting information about their family tree, so you can make a connection, so you can possibly break down a brick wall. Okay. Back in 2010, I wrote a paper about my experience with mitochondrial DNA testing. And I contacted several other matches. Their matches either 2 steps away, or in one case, 3 steps away. And as a biologist and an artist, I wanted to find out to see if there was any family resemblance at all, because many times when we go to get-togethers or family reunions, we'll say, "Oh, you look like Uncle Jimmy," or "You look like Aunt Sally." Those family traits go and are very well recognized by people. People are really good at recognizing similarity of family members. So what I did is I put together a composite graphic that shows family A I think you recognize that my family is my family A. Family B, family C, and family D. Now, family D, this lady in the picture right here, was adopted and didn't know her heritage or ancestry. Lives in the United States and didn't know essentially that she has potentially, she definitely has European ancestry on the maternal side. The question now is that can we determine by just looking at some faces to see which ones are 2 steps away and which ones 3 steps away from me? And because I don't have a live audience that can interact really well, I will basically tell you that family B to my family is 3 steps away. Family C and family D are 2 steps away on my maternal line. But family C and family D are exact matches. Now, do you see any commonalities between facial features, and this is also genetics as well, between different families? Let me point some out to you. Oldest family member that I have a photograph of, and another family, she's from Poland. Family B is Northern Ireland. So one of the things that I look at is bone structure on the face, because the skull is something that does not change very quickly and has genetics that stay around for a long time. What I noticed right away is in the eyes of family B, and the eye shape, the eye socket shape over here is very similar to my great grandmother in family A. Not sure if you see that or not. But what I also noticed is that my grandmother, Wanda, her high cheekbones over here and face and eyes look very similar to family C over here and family C and family D over here. There's a lot of resemblances over here regarding that. What's interesting though is that Wanda's sister right here looks a lot like the sister that's over here. So I contacted family C and I said, hey, I'm gonna send you some photographs and things like that. And for a couple of weeks, I didn't hear back. And I was like, oh, this is going nowhere as far as my research goes. And I sent a reminder email, hey, what'd you think? And I got a quick response that says, oh, we're definitely related. I'm like, okay. So I went and did more research on that. And it seems like we were related in the 1700s with family C, my family. Family D and family C are even more closely related for a timeline. But family B and my family are probably related closer to the 1500s. And I go into more detail about that, but I'm not going to do that right now. So we're on time. Okay, there are other companies that provide paternal and maternal DNA results, including 23andMe and Living DNA, but they're not as easily researched. They'll just give you your genetic group, and that's pretty much about it associated with that. There is some raw data you can take, but it is much more difficult to access and actually manipulate, so, or use to compare to other people. 23andMe provided me with my genetic maternal group and my paternal group, and kind of give some indication about origins, age 26,000 years ago for H2A2A, and my R group about 27,000 years ago in Eastern Europe, Scandinavia, and Southwestern Asia and India. That's where they're found. Living DNA gave me some images of what look like people. a maternal line and paternal line YDNA of R1A. It's giving me the last twig. They're saying CTS 3402, which is close to what family tree DNA has gone even further on mine. And then my haplogroup H is H2A2A1, and they got that right. Initially, they got it wrong. They got it. They thought it was some group, maternal group from India. And I told them basically 3 other companies were saying it was totally different, and they went and checked. They said that my DNA was so close to the Cambridge reference sequence, which is what I said that lady who donated her DNA to science for that 1981 full sequence results. I'm so closely related to that it skewed their computer system a little bit and read to a different group. But they corrected that after I asked them to. Other resources you can go to regarding genetic groups, Wikipedia and Upedia have a lot of information and maps that show where these genetic groups are located. Kind of gives you some more information if you've done testing with them. Okay, I believe that's about an hour. It takes me a little longer to get through this presentation. If there's any questions, I'll be happy to entertain them.
Okay, I have a few questions here. The big Y results is only for men, question mark? Is that correct?
Yes, that is correct.
How can it be useful to women connecting men? How can it be useful to women?
Okay, the only way to be useful is if you can test a brother or uncle or father that is your direct relative. You can even go to a cousin, but you have to make sure you follow the path that is on that one male line. But don't go too far back just in case there might be some differences in paternity and things like that. So you just never know. I'm not saying it is, but I'm just saying you just never know sometimes. But if you were able to, now there are some people that say, well, I don't have a living brother anymore. I don't have a father anymore. The father's passed away too. Try to find an uncle that had, or another either half sibling or full sibling on the father's side, or a uncle that has another cousin that you can contact that is a direct descendant of that surname that you know of, and then you can use that. You don't need to do the big Y for paternal YDNA testing. Start out with the 37 or 67 marker test, and then move up if you want to go any further advanced than that.
Okay,
Okay,
Tricia,
I think that answered 2 questions from 2 different people then.
Okay.
So then another one is, with the mtDNA matches, does it give you names also, or just the push pins?
No, it gives you names of the person who is tested, and then if they put down their location, it'll show them on a map. You have to go to a certain section of that your results page to get the maps. I did that just to make it a little easier to see, but there is a list that you get, and then in each list there is an email icon that you can email that person. So you can share information to see if you can find a common ancestor.
Tricia, I caught one. There's a question here. I've noticed emphasis on using paternal and not paternity. Could you elaborate on the significance?
Yes, a paternal YDNA test is strictly the Y chromosome. It is following the paternal line. A paternity test follows paternity of, let's say, a couple has a child. They don't know who the father is, or want to make sure that the mother is the mother. That is an autosomal test following a certain specific markers that are used to determine paternity. And that is a separate test, does not look at the Y chromosome, but looks at the autosomes, the rest of the DNA at specific markers to see if they match up, because they're looking at both the father's side and the mother's side for that one child. Whereas a paternal DNA test is looking at that one long line from the father to the grandfather, great-grandfather. I hope that makes it more clear.
I've got another one.
Sure.
Do we need to understand everything you've described to get results? Or do the site's own results page give the full results?
I'm gonna stop sharing here. Your test that you get, well, I should say you take the test, you get the results between 2 to 6 weeks. A lot of the testing companies have provided a number of things to read regarding your results. Some have tutorials that are either slide presentations or videos that you can follow to better understand your results. These presentations today are to give you a first step into understanding some of the things regarding your DNA results, what you might be able to expect and what format they would look like in, so that it becomes more familiar to you. Repetition of seeing these things over and over again will make it a lot easier to understand and get the most out of your DNA results. So the companies are doing pretty good things regarding additional information. In fact, some of the best written small articles I've read to expand my understanding has been through 23andMe, where they have little offshoots of things that you might want to know more about, and then there's a little bit of reading involved, but they are pretty good about that. Now, one thing I will say is I wouldn't go into too much on their help desk or anything like that. That might be a long wait for something like that, especially since so many people have tested now. But typically, a lot of the questions are in there in the FQA or FAQ. I'm sorry, frequently asked questions, and they do a pretty good job. If you have any questions, you can always email me for a quick question. Anything else?
There's a question here. In the afternoon session, will you get into analyzing matches, centimorgans, segments, etc.?
Yes, we'll be looking at the next presentation is about autosomal DNA.
Tricia, did you catch any others?
There's one, and maybe I missed the answer. With mtDNA, you use steps rather than generations. Could you please elaborate more on what a step is?
Okay, the step is not a generation. The step is a mutation or change in the DNA. The generation is basically explained by a time frame of 25 to 30 years between generations, but a step is essentially if you have a difference in the DNA from someone else. It depends on how many differences you have or mutations. Then instead of calling them mutations, some companies have called them steps. I did see one about genetic engineering. We're really not going to get into that, but there are tools like CRISPR that is changing the face of a lot of things, but that's a tool that's available globally, but there are ethics issues and things like that that are making things, especially in the United States, a lot more legally oriented than others. So that's beyond my purview. Anything else?
I think that's all the questions we have, unless Amy, you have any from your in-person group there.
I do have one from the in-person group. Can you hang on just a second?
Oh, sure.
Okay, go ahead.
What time is the next,
Okay, we'll take a lunch break in a minute. The next one will be at 12:15. Would you like me to
12:15.
just, I can get his question and come back to it a little later if you like.
no, I'm okay.
Okay, go ahead.
Can I significantly, with the knowledge that I'm learning here, and with
I can't hear him.
tribunal
You're gonna have to repeat the question.
What I know with DNA, can you see me? Hello, hi, my name is Tyler. What I know with DNA is they come on a spectrum of color. That's what I know. Can I alter my chromatic braiding, is what they call it, through like tribunal practice with what I eat? Let's say that I want to change from being a carnivorous or omnivorous to purely fruit, vegan. Will that change significantly my DNA in the line?
It's outside the scope of this, but I would say no, it would not. Your DNA is pretty conservative regarding changes. The changes happen when it is replicating in your body, but it is like a copy machine and tries to keep that copy as close to as possible, because if there are things that go wrong with the copy, there can be deleterious problems that could arise because of that. But I would say at this point, the difference between being a carnivore and a vegan is more a decision of personal choice. At this point, that's as best as I know.
And what about stimulations? Are there altering stimulations, or can I alter stimulations in my life that would alter my DNA?
Not that I'm aware of.
Okay, thank you.
Sure.
Okay, I have one more and then we'll take a break. How do they know your most distant ancestor's name? Is it from someone else's family tree?
The first thing you do is you find out if you have a common ancestor, and then determine the time frame for that common ancestor. The only time you can get a name of a person is if you compare family trees.